All right. So welcome everyone. So today is a little bit different. So Dr. Huth encourages his teaching assistants to try and do a guest lecture, and in true student fashion, I kind of waited till the very end to figure out what it is that I wanted to talk about. But once we get started into today's topic, it's gonna kind of be clear that this would have been best placed as sort of the second to last, the penultimate type of lecture, but unfortunately, I'm out Friday. That's gonna be my last med school interview, the very last one. And so to anybody who ends up joining on the Zoom, I know that there are others out there who are going through the application cycle as well. So if that applies to you, I'll say really quickly, briefly, I hope that you've received good news. I hope that you continue to receive good news over the holiday break, and that if you don't, I hope that you're able to look back at this time and still view it as good news. But yeah, other than that, we can get started with today's lecture. So a couple of months ago, my certificate and research mentor, Dr. Joshua Chang, had invited me out to one of the lab meetings. And I was sort of under the impression that this was an undergraduate heavy lab, but when I came out to the lab meeting, I ended up being one of the only undergraduates there. It was a room full of physicians and professors. And as the meeting went on, it became very clear that I was absolutely useless in this meeting. It's nothing like It's a Wonderful Life. The world and the meeting went on fine without me. But as much as I felt useless, I also felt inspired, because I think I got to at least sit in and watch experts of their field collaborate with each other and contribute their insights. And however minimal my work was, it still felt like it was contributory to something larger than me, and it was that feeling of inspiration, that feeling that what we are doing and learning about is relevant and important to the goal of that feeling of that what we're learning is relevant and important. I would say that is the goal of today's lecture, because over the past semester, let's see if it goes. Yeah, so that's the goal of today's lecture, because throughout this entire semester, you guys have not only been learning programming concepts, but also practicing programming skills, which means learning about the ins and outs, learning about the details, learning about the minutia of how it is to get Python to do what it is that you want it to do. Today's lecture is not going to be that. Today's lecture is going to be big picture. I'm going to go through each of the 6 units that are listed on the course schedule and try to point out a cool example or a cool application of it being used in the real world, and why these skills that you guys learned were vital to that cool concept or project. Any questions so far about where we're going today? Okay, cool. Let's see. Okay, so for the first quarter of the course, we talked about variable types, arrays, indexing them, slicing them, all the fun Python and NumPy basics. It's something we've gone back to throughout the entire semester. I mean, we just did it again on this past Monday's quiz. And the reason is it's really hard to understate how basic and how important it is to be able to do data analysis with Python. The bread and butter of research is that data munging, as Dr. Huth likes to call it, right? So how do we use these Python and NumPy basics to manipulate data? For example, if we had if we had these EEG readings from multiple trials, you guys now know that you guys can store that as a 3D cube of data. And if I told you guys, I just want to look at the 4th electrode reading and look at that recording for the entire duration for all the trials, you guys would know easily how to pull that out. It's just array 3, comma, colon, all the columns and all the layers. So that stuff, very basic, but very fundamental to being able to do data analysis with Python. Then the next unit we ended up looking at was Matplotlib and the principles of data visualization. I don't have too much on this slide because I feel like we've already talked at length about all the harms and ways that you can visualize data in a misleading way and in an improper way. But yeah, I just wanted to give the book that Dr. Huth read a quick plug again, The Visual Display of Quantitative Information, that explicitly pointed out all the best principles of data visualization. Up until this point, do we have any questions or? Okay, cool. Moving on, after the data visualization unit was the probability and statistics unit. So statistics is probability and statistics. This is the math of uncertainty. And so even when we don't have access to all of the world's information, we can still use samples to make valid conclusions about the world at large in a scientific and logical way. So here's an example. If you guys don't remember from Neuro 335, a connectome is a map of all that shows the connections and wiring of all the different neurons in the brain. So if you're somebody who studies these connectomes, a natural question that you may have is, which areas of the brain coactivate? It's a natural question because it's a Hebbian principle. If neurons that fire together wire together, and so if neurons are firing together, that usually means something. It usually means they have some sort of shared purpose. But I would say, hold on a second, because if they're completely synchronous, meaning they're completely coactivating completely at the same time, that's usually kind of suspicious, not meaningful, and is usually going to be associated with some sort of like seizure-like activity. So completely synchronous coactivation isn't a great idea. But if they're too out of sync, then how do you know that you should be grouping these brain regions together, right? So a more scientific way to phrase this question is, which areas of the brain coactivate to a statistically significant extent? And during our statistics unit, you guys know the consequences of how we define statistical significance and when it is appropriate to adjust our thresholds, and how do we do that in a logical way without hacking the statistical threshold, the alpha. But whenever we did figure out ways to group these brain regions together in a logical and statistically significant way, we ended up getting these groups, these lists and collections of brain regions that seem to fire together in a functional way. If you're in connectomes, that term for that is called a network of brain regions. And we ended up finding seven major brain networks upon which other minor networks seem to align themselves with to carry out some executive functions. Some of these networks include the default mode network. So the default mode network is basically the region of the brain regions that are active, not when you're resting, but just when your brain isn't actively participating or engaging in anything. So me on Sunday church. The other networks that we have is the central executive network, the salience network, and there are other different types of networks. And it's been theorized that aberrations and anomalies in these networks are what cause some of the more prominent neurological and psychiatric symptoms that we observe. Any questions about connectomes? And we would not have been able to group these in a logical way had we not known the importance of statistical significance, which is something that you guys learned about in this unit. Any questions up to this point? Okay, cool. Really quick fun fact that I thought I should mention. So when I took Neuro 335 in spring of 2022, the most complicated species for which we had a completed connectome was C. elegans, the flatworm. I think it was like 302 neurons. As of last month, we have just completed the connectome for Drosophila melanogaster, or the fruit fly, which is basically the poster child for a lot of behavioral neuroscience experiments. And so the fact that we've been able to accomplish this convinces me, and I hope it convinces you, that neuroscience is one of those budding and emerging frontiers that is quickly growing and expanding. Oh, and something else I found interesting. This is a recent textbook by a couple, I think one of the, Jackie T. Young, I know for sure is a neurosurgeon. I don't know the credentials of the other authors, but this is called Connectomic Medicine. Basically, they are hoping to use connectomes and investigating and targeting specific neural circuits to improve neurologic care. So if that's something you're interested in, something else that you might want to consider checking out. All right. So after our probability and statistics unit, we ended up moving into our time series unit. So in our time series unit, we've talked about time series and how signals are a special type of data because of their oscillatory nature. So they go back and forth. We have these negative feedback loops. We learned how to break down a signal into its component frequencies. We learned how to convolve a signal with another signal so that we might be able to filter out certain frequencies to prevent artifacts in our data. And some examples we discussed last time of signals in neuroscience are EEGs. And I had a Canvas announcement about the stroboscopic effect. Basically, the stroboscopic effect is if you're looking at a ceiling fan or a rotating fan, and you start to notice some examples of signal processing in neuroscience. But one neuroscience example where I think signal processing is incredibly important is in the world of electroceuticals. So electroceuticals are a play off of the word pharmaceuticals because when it comes to using drugs to correct the brain, we've kind of had a mixed bag of results. So the whole idea behind electroceuticals is to be able to use electricity and deliver the right amount of electricity in a therapeutic way so that we can stop pathologies like epileptic seizures, like cardiac arrhythmias, like Parkinsonian tremors. The problem with electroceuticals, though, is that it requires very, very precise timing. And so what that entails is that you need very accurate instrumentation. You need to be able to make sure that you are measuring correctly all of the voltage and electrical fluctuations that are happening within the brain. The other thing that you need is you need to be able to process the signal quickly enough. So being able to process the signal quickly enough so that you know at what point in the oscillation is the brain state currently in, so that you know exactly the correct time that you can deliver the impulse, and you have to be able to make that calculation very, very quickly. These provide some of the reasons why the precision of it is probably making it a little bit impractical, which is why one of the frontiers of research for this has been phase agnostic stimuli. So what does phase agnostic stimuli mean? It's basically trying to find the right shape of the electrical stimulus. So instead of a square pulse, maybe something a little bit more creative. And the goal with that stimulus and finding the right shape of stimulus is so that we are able to counterbalance whatever oscillations is going on in the brain without having to wait for the right phase. We can do it regardless of what the phase is. That's why it's called phase agnostic stimulation through finding the right stimulus shape. The cool thing about this is that the people who are leading this research is Dr. Joshua Chang and Dr. David Pedafar, both researchers at UT Austin. Dr. Joshua Chang is the guy that I mentioned at the beginning of the lecture. Love the guy. And Dr. David Pedafar is currently, I believe he's the department head of neurology at Dell Medical School. And so it's glad that this kind of research is happening on the 40 acres. And it's an example where signal processing and knowing how to extract information from signals becomes incredibly important. In fact, there's a whole textbook for signal processing just for neuroscientists by Dr. Drongelin. And I believe he has a accompanying YouTube series. So he has an accompanying lecture series on YouTube, and I think he mostly uses the chalkboard though, so I don't know how you guys feel about that. But any questions up until this point? Yeah. Yes, yeah. I don't know if it's the DBS is in that realm of electroceuticals. I don't know if that's the exact one that they used, but I haven't looked at the paper in a while. So yeah. But yeah. Any other questions? All right, great. I realized I was talking really fast, so I can slow down a little bit. But the next unit we moved into is machine learning. So coming back to the realm of statistics, once we've gathered enough data, we want to be able to predict what future data should look like, and we can do this using machine learning. There are 3 use cases of machine learning that we've talked about. The regression model one is the one that we've talked about the most extensively. When we talk about regression models, we're talking about fitting a linear regression so that we minimize the error functions. We talked about cross-validation, which involves breaking up your data and withholding certain pieces of your data set so that, one, you can fit your linear regression to estimate your parameters, evaluate those parameters, and then finally be able to test those parameters against your third set of data. And all the while, we want to be able to regularize the data, because if it turns out the 2 things that you're analyzing, the 2 features you're analyzing happen to be somewhat related, there's a good chance that you might accidentally overfit the data. And so regularization prevents that so that those parameters don't explode unnecessarily. So that's the regression model we've talked about. We recently talked about classification, which is an unsupervised machine learning method. Basically, you don't know. Sometimes we do know. Actually, it can be supervised or unsupervised. So sometimes we know what category the data is in. Sometimes we don't. And so classification is just basically how do we categorize and figure out which category a data point belongs to. And then we last left off on Monday with a dimensionality reduction technique known as principal component analysis. A dimensionality reduction techniques are so that if you have data points with lots of different features, a lot of different values and data associated with that data point, we want to be able to find what is most salient and be able to reduce that dimension, those dimensions to just the important features. And also because for visualization purposes as well, we only work in 3-dimensional, we can't see anything really above 3D or can't visualize it. And so that is another use case for dimensionality reduction. So that is sort of the 3 use cases of machine learning that we've talked about so far. Any questions about that point? Okay, cool. So all of these machine learning techniques was what the Mayo Clinic used in 2022 to create a computational model of Alzheimer's after analyzing a bunch of PET scans. The PET scans were looking for a specific type of glucose. I forget what kind of glucose it was. But for context, when you're using a dimensionality reduction technique like PCA, you are looking for the principal components. You are looking for the axes with which capture most of the variation in your data. I think the analogy used was potato. We want to see how can we capture most of the variation in that cluster of data. If you ever plan on taking linear algebra, the way that you calculate this is called calculating something called an eigenvector. And in this particular paper, they used a different dimensionality reduction technique, but they were able to go from 150,000 dimensions for each of the voxels in the PET scan, and they were able to reduce that down to just 10 dimensions, and they called these 10 dimensions eigenbrains. They were able to figure out that these eigenbrains typically represented some sort of processing ability in the brain. I don't totally know what each of the eigenbrain axes mean, because it isn't just one voxel, it's a combination of different things. So for example, if we look this is on figure E, you can tell that they plotted out the linear combination of the first 3 axes, or the first 3 eigenbrains. The first eigenbrain one is called feed forward versus feed backward, or feedback. I don't really know what that means. The second one was abstract models versus concrete models. And the third eigenbrain axis was external data versus internal data. And so that cube that you see there is a linear combination of those 3 eigenbrains. And they plotted out where in that linear combination you can find different cognitive abilities, like not cognitive abilities, but cognitive capacities. So object recognition, social recognition, reward. Those are some of the things that you could predict using just the first 3 of the 10 dimensions that they reduced it down to. They realized that reducing it down further than 10 dimensions didn't really get them anywhere. But the model that they created was so good that it was able to make valid predictions about the extent of a patient's neurodegeneration in 7 other disease cohorts. So these cohorts included just the normal aging population, the cohort of patients with dementia, with Lewy bodies, patients with posterior cortical atrophy, and another cohort who had a variant of primary progressive aphasia. But those were some of the other disease cohorts for which this Alzheimer model seems to apply and seems to be predictive of. And so, again, this model that they created was based off of using the previous machine learning techniques that you guys were all got familiar with over the past couple of weeks. Any questions about this particular unit? Yeah. Hmm? Yeah, so I don't remember the legend for figure A, but both of them, they're just looking at that second axis, eigenbrain 2. And let's see, I don't know what they're defining as functional connectivity, but it seems that depending on where along that axis of eigenbrain 2, you can start to detect where certain abilities in your brain are associated with. So if you go way along, you go all the way to the right of that eigenbrain 2 axis, you'll find areas associated with, I believe that color is corresponding with social cognition. And then if you go all the way to the right, and I think this is abstract models. So very abstract is social cognition. And then very concrete would be, let's see, blue is motor function. So very concrete. Yeah, so that's what figure A and B mean. I wish I could give you a better definition of function connectivity, but I don't.
So,
yeah.
The functional connectivity here, this is actually called for the principal rate of functional connectivity. So, it's more or less just the first principal component of like brain.
Yeah. Any other questions about the Mayo Clinic model for Alzheimer's? Hmm?
They're able to find changes in cognitive abilities.
Yeah. Mm. Yeah, I would say so. The bar for getting anything that's like statistically significant to we got the thumbs up for clinical application. That's like a very long process. But my hope would be that sometimes the diagnostic criteria for not just neurology, but also psychiatry can be very vague. I think currently in psychiatry, I don't know if you've heard the diagnostic statistic manual. It's literally just a checkbox of things that you check off. If you agree to these, if you feel like you have these things, then you're eligible to get diagnosed with that condition. I hope that the use case for a model like this is that we don't have to be so subjective with our diagnostic criteria. We can put somebody through the PET scan and be able to more predictively, accurately categorize what kind of dementia or what kind of neurodegeneration they have. And so I think that would be the hopeful use case of this type of model. Yeah. And I would hope so too. Personally, I think neurology and psychiatry are very subjective, and you know, some people, I've met a couple of psychiatrists. Some psychiatrists are very like, go with the flow, you know, that's life kind of psychiatry. I'm more interested in the type of psychiatry that seeks to make this kind of more concrete. And there's a textbook I'll refer to later on about that. But yeah.
Yeah. I'm broadly kind of skeptical about this.
Yeah.
About like, I think there certainly are underlying biological bases for psychiatric disorders. Like that's why they exist and why they come in like certain symptom clusters, whatever. a lot of our current techniques for trying to get like biological truth
Mm.
in
Yeah.
psychiatry is like, it's just messy. It's like
Yeah.
really messy and
Yeah.
it's expensive and imprecise.
Mhm.
And compared to this gold standard of just like asking somebody some questions about
Yeah.
their life,
Mhm.
like anything we can do with neuroscience still kind of sucks. That's really
Yeah.
like there's no way to solve this, but it's like
Yeah.
for the last 40 years, people
Yeah.
have been trying really hard to
Mhm.
solve this.
Yeah.
And not actually getting very far.
Yeah.
It's overall, like pretty disappointing for the fields. There was a big retrospective on kind of like the failure of fMRI in particular to like penetrate into medical practice. Like fMRI is not used in any diagnostic test.
Yeah.
Currently, and it's been around for 30 years.
Yeah.
People have been trying for 30 years.
Yeah.
Like, really hard to make this happen.
Right,
And it's
right.
like
Yeah.
hasn't really happened. It's a little depressing, but it's like
Yeah.
I don't know. It also speaks to I think the power of really just talking to someone.
Yeah.
And seeing,
Yeah.
what do they feel about themselves?
Yeah, yeah. The social cultural context of it is really important. You can't pick that kind of thing up in an fMRI. I would say I had a very similar experience of disillusionment when I'd heard about this thing called so as an alternative to the DSM-5, which is just a list of checkboxes. The NIMH came out with the research domain criteria, which is they provided a framework that's like, hey, let's make this thing a little bit more objective. But there are but I did read a criticism of the research. First, I had shouted out a psychiatrist who said, yeah, I'm not a fan of it because it's just not how we practice psychiatry. And then that let me look into it, and somebody had published a paper on the criticism of our doc, and it relied on some very faulty assumptions of like, it sort of assumed that psychiatric conditions are on the spectrum, and that it assumed that psychiatric conditions must be on this spectrum. Like, if this is normal and this is pathological, psychiatry must be somewhere in this spectrum. But the reality is we don't even know if that's a correct assumption. We don't know if some people really like the disease state model, which is this is normal, this is pathological, like this is disease state, and that's how we'll get better treatment, not by pretending that everything is a continuous spectrum. And it's a faulty assumption at best. So yeah, I had a similar experience of disillusion. Okay, maybe this is a lot harder than it looks. But I will reference a textbook that was published in 2020, Computational Psychiatry. And I think the authors are optimistic, and I think it's still a field. I personally hope it's still a field that's ripe for improvement. But yeah, it does seem I hope it's a field ripe for improvement because the past implies it's been pretty disappointing. So yeah. Any other questions on this topic? Okay, cool. So the smallest and the last unit we had on the course schedule was the neural networks unit, which I think is going to be the one that we unfortunately run out of time for. But neural networks are just another supervised machine learning technique that has proven to be incredibly powerful. It's been a technique that's been incorporated into all sorts of pattern recognition tasks, large language models, and artificial intelligence. In fact, Dr. Huth has leveraged ChatGPT in his work in order to create a model that predicted a stream of speech just based off of somebody's neural activity, which earned him a coveted CNN coverage spot. You guys can look up the headline anyway to figure out to watch the video. But yeah, there is another interesting use case of neural networks that I wanted to touch on briefly, which is that all of the machine learning models, when we talked about models throughout the course, right? We've been referring to these models, we can typically categorize them as data-driven models. So in other words, they are statistical models that are formulated based purely off of the data that we feed them in order to make predictions about the data, right? However, there is another category of models called theory-driven models. These models attempt to simulate or replicate some neural process based on what we know about the brain. Three types of this. So this is the textbook I referred to earlier. is computational psychiatry. It's a compilation of different authors about, but I think it's organized very well. And the contents are organized very well. And in its opening, it talks about these data-driven models, but it also talks about these theory-driven models, right? And so 3 types of theory-driven models that have shown a lot of promise, relatively, include reinforcement learning models, Bayesian decision theory models. And so if you liked, going back to our probability and statistics units, if you really liked the idea of probability and statistics, and you took a look at the course packet that I put out, if you were really interested in that, Bayesian statistics is something else that you could be interested in. But yet again, neural networks makes another reappearance, not only just under machine learning data-driven models, but also as a theory-driven model. And the reason is that they're not just a machine learning technique, it's a machine learning technique that was inspired by biological neurons. And so if we can incorporate the correct neuronal firing dynamics, which is a totally different subject. So that think like Hodgkin and Huxley type model, integrate and leak, integrate and fire model, leaky integrate and fire. There's another one, Fitzhugh and Nagamo. Those are all different differential equations that describe the dynamical system that is neuronal firing. It's not, I know in neural 1 we can't teach you, if it goes above the threshold, it fires. It's a lot more complicated than that. But if we can incorporate the correct neuronal firing dynamics, and we can think critically about the connections we make in the network, the neural networks are actually getting better and better about approximating real neural activity. So in a kind of roundabout way, neurons inspired neural networks, which are now trying to replicate neurons again. And so that is another use case for neural networks. Any questions up until this point? Okay, cool. So now we've talked a lot. We've talked around neural networks. I think we have a bit of time to actually define what a neural network is. So what are neural networks? They are made up of individual units called artificial neurons. These are also called perceptrons. And so what is a perceptron? A perceptron is nothing special. All it is a generalized linear model. That's it. It's just a GLM. So you can see that on the left side, the perceptron will take in a list of values called inputs. It will multiply each of those inputs by some number. We call it a weight. You add it up. You add a bias. You add some arbitrary value that you can add on to there, and you get a number. And that's it. That's all the perceptron does. The thing that may distinguish a perceptron from a GLM is that after this sum is obtained, the perceptron will pass it on to something called an activation function. And so this activation function will ultimately determine what value the perceptron is going to output. Some activations include a sigmoid function, which is, I think if it's beyond a certain value, it's going to fire a 1. If not, it's going to output a 0. There's also something else called a ReLU activation function, which I think is if it's just above a certain, actually, I don't know. Do you know what a ReLU function is more specifically?
Yeah.
Yeah.
So it's basically if the value is positive, it just gives you the same value. If the value is negative, it gives you 0.
That's right. Yeah, yeah. So, oh yeah, it's like that line. Yeah. So after that, after the perceptron passes it through the activation function, you get an output. And so ultimately, this is just a fancy generalized linear model where you have a bunch of variables, you multiply those variables by some scalar called the weight, you add a number called a bias, you add that up all together, you pass it through an activation function, and you get some other number. So ultimately, you have an input of a couple of numbers, and you output a number. In this example, we output just the one number. Actually, I think it will always, yeah. So you have inputs, you input values, and then you output a value. That's all an artificial neuron is. That's just the perceptron. And from that point, all you need to do to build your neural network is to connect a bunch of these perceptrons together. So these perceptrons can take inputs alongside other perceptrons. So you can see that these inputs are feeding into other perceptrons, but in a parallel fashion, right? The other way that you can connect perceptrons is to layer it. So the input from the output of one perceptron passes it on as the input to another perceptron. In this one, there's just the one layer, but if you can imagine sort of this copied and pasted, this is just another layer where the output of the perceptron becomes the input of the next perceptron and so forth. And ultimately, that is what happens, is that you have a list of inputs and you get a list of outputs. And then from that point on, it's just like any other statistical model, where if you have a set of data points, known data points, where you know what the input is, you feed in the input, adjust the parameters, adjust the weights until you get that desired output, and then do that again with the next pair of input output. Do that again repeatedly so until you've adjusted the weights just so that it is able to reasonably predict all of the outputs that you obtained from the inputs, just like you would train any other statistical model. And that's basically what a neural network is. Any questions about what a neural network is other than a fancy statistical model?
Another way to think of it is also just like a fancy regression model.
Yeah.
So like it's a nonlinear regression model. It's a regression model that takes like feature inputs, like what a regression model would, but it's not restricted to like the output being aligned with respect to the feature inputs. The output could be like output could be anything. And it turns out that's all you need to, I don't know, simulate brains.
Yeah.
To feed ChatGPT. It's kind of nuts.
Yeah, so that's ultimately what it is. And that's another way of thinking about it. Sometimes people will tag on labels. So this is just one layer, but if you imagine another layer and then another layer and another layer, and then if you have enough layers, you can call this a deep neural network, deep learning, which is just, again, machine learning is just another way of training and predicting statistical models. Neural network is just a fancier version of that. Deep learning is just a fancier version of that. But yeah, let's see. But yeah, you just train and adjust it. You adjust the weights just like you would a statistical model where there's a loss function, whether it's gradient descent or some other loss function. You minimize that and repeat that until you get your neural network starts outputting the correct outputs that you expected for those inputs. Any other questions about this neural network? Cool. So because we are just kind of calculating and passing along values in a very straightforward fashion, and we calculate values and pass it in the forward direction, right? We can call this a simple feed forward network because it's just feeding values onto the next layer of perceptrons. But that actually isn't there is another type of neural network that is arguably more biologically plausible. Our brains aren't just completely linear where we have input and output and that's totally it. We also know that our brain abides by Hebbian principles, which is what I mentioned earlier. It's the idea of firing together and wiring together, right? So this is one example of a neural network. There is another example of a neural network called a Hopfield network. This next type of neural network is very different, but instead of having input and output values, it has input and output patterns. So it's a little bit more complicated than the feed-forward network, but here's how a Hopfield network works. Neurons are either activated or deactivated. They are represented as either positive or negative one. And their activation status is determined by the activation status of all the other neurons. You can see that in a Hopfield network, every neuron has to have a connection with all the other neurons. They have to be all connected. So obviously not every neuron in your brain is connected to other neurons, but that is the rules for a Hopfield network. It has to be what is called a complete graph. And so with this, the activation status of a neuron is determined by the activation status of all the other neurons multiplied by some weight, multiplied by some connection. And you can see that the weight is bilateral, right? So at first, in the feed-forward network, it was just one direction, whereas here you can see that whatever weight is assigned here will apply to both this neuron and the other neuron. So it's bidirectional. Because information flow forward and backward in this neural network. This is called a recurrent neural network instead of a feed-forward network. But yeah, the neuron's activation status is determined by the activation status of all the other neurons plus some weight. So let's say I have updated the weights and trained the network such that it recognizes any pattern that remotely resembles this pattern, right? So here's the pattern. Neuron 1 is on, neuron 2 is off, on, right? So say that's the pattern we're trying to recognize. Then, and I strongly train the model, like the weights are very, very strong. So if I were to present this if I were to present the network with the same pattern, present the network with some stimulus that causes the network to assume a pattern where this is on, and this is on, right? This is on, that's off. So basically everything's the same except for this one neuron, right? The weights of the activation status of the other two neurons and the weights of those those connections will force this neuron to turn off, right? So it will flip back. So if I made a pattern that's very similar to this, except this one is on, the weights multiplied by the activation statuses of the other two neurons will force that neuron to turn off, so that it recognizes that this pattern resembles the pattern that we trained it on. Does that sort of make sense? So it's not input values and output values, it's input patterns and output patterns. Does that sort of make sense about what this network is trained to do? It's very different than a feed-forward network.
What did it turn on to? This is the simplest recurrent neural network.
I don't know if it's I feel like not the simplest recurrent neural network. I feel like there are feed-forward networks that probably have a connection that loop back to the first layer or something. So that would be defined. Yeah.
This was the first like artificial recurrent neural network.
Yeah.
That people really characterize in any kind of detail. Nowadays, recurrent neural networks are much fancier and bigger and do all kinds of crazy things. There's recurrent neural network models that are not quite as capable as ChatGPT, but are similar. And this is kind of a granddad of those. Anyway.
Yeah. But yeah, so that is one of the simplest recurrent neural networks that became recognized as being potentially useful. And so, for example, this is a Hopfield net with 25 neurons. Obviously, if they're all connected together, it's kind of hard to tell where is where. And so you can abstract this as sort of a pattern as a 5x5 grid. 5x5, am I counting that right? Yep, yeah. So 5x5 grid. And let's say that this is the pattern that you train the network to recognize, right? This is the pattern, and where the blue neurons are on and the white neurons are off, right? So then if you present some sort of stimulus that causes the network to light up in this manner, it will still be able to recognize that pattern as this pattern. It will force this neuron to turn off, these neurons to turn on, and that neuron to turn on as well. Does that sort of make sense about the pattern recognition that Hopfield nets are capable of? And so it doesn't have to just be one pattern. You can train a Hopfield net to recognize multiple patterns because the only difference is that the weights become a little less strong because they have to be adaptable enough to recognize when one pattern looks like this pattern and when one pattern looks like another pattern, right? So you can train Hopfield nets to recognize multiple patterns. One of the drawbacks is that it's not very efficient and there's a limited capacity. There's only a maximum number of patterns you can train the Hopfield net to recognize before it is unable to detect when it is unable to separate some patterns from other patterns. So there's only a maximum number of patterns that you can train the Hopfield net on. And so this represents that. In Hopfield networks, you don't really have an error function because you're not comparing known outputs to the expected output. You're not comparing outputs to expected outputs. What you're comparing it to is energy is kind of hard to define, but in simplest terms, it always seeks to minimize the energy so that it resembles the pattern that it was trained on, right? So for example, if you presented a pattern that's somewhere up here, right? It's going to go back and forth. Some neurons are going to flicker on, some neurons are going to flicker off. But ultimately, if it doesn't start resembling the pattern it was trained on, it's going to end up in one of the spurious minima, right? So that is a pattern in which you didn't train it on, but that's just the pattern that it ended up settling on because the pattern you gave it was too different from the patterns you trained it on. But this global minima, that is the pattern you trained it on. And so any pattern, any state that remotely resembles this pattern that you trained it on will flicker and go back and forth until it ends up resembling the pattern that you trained it on. Does that sort of make sense about how this energy landscape describes how a Hopfield network will try to default to the patterns that you trained it on? Okay, cool. And so the con that I mentioned earlier is that it's not very efficient. It has a limited memory capacity. But the pro is that it really embodies that associative Hebbian learning, that the idea of neurons that firing together or wiring together, much like a brain. There is a biologically plausible explanation for Hopfield networks being in use, being put to use in our brain. So for example, you can think of this pattern, this heavily trained pattern, as a super vivid memory from your childhood, right? Something super distinct, right? And so anytime you get a stimuli that remotely resembles that vivid memory, your brain will start to trigger that memory of that vivid childhood memory, right? And so that is an example of a Hopfield network being put in a biological context of being able to remember super vivid memories, and those vivid memories being triggered by stimuli that resemble the vivid memory. Does that sort of make sense, or? Anything else that you wanted to add onto Hopfield networks, or?
There's, even though Hopfield networks are kind of like venerable, they're old in neural networks classes, they were unusual to talk about probably until this year because John Hopfield won the Nobel Prize for it, which sorry.
Yeah.
But there was a big development a couple of years ago in 2020 where some people discovered that actually what Hopfield networks do is very similar to the neural network mechanism that underlies something called the transformer network, which is what ChatGPT actually is. So Hopfield networks are maybe more like this very modern thing, and some ideas from Hopfield networks apply to this modern thing. The ideas from this modern thing also apply to Hopfield networks. There's a really cool like convergence of neural network theory that happened recently. So they're more relevant than we thought maybe 10 years ago.
Yeah. I remember reading the seminal his capstone paper on Hopfield networks. It's very old, but yeah, it's making a comeback. So much so that the Nobel Prize in Physics for their contribution for foundational discoveries and inventions that enable machine learning with artificial neural networks. John Hopfield and Jeffrey Hinton were awarded the Nobel Prize in 2024. But you guys don't need a Nobel Prize to know that the work that you guys have been doing throughout this class has been meaningful. So that is it. That is all I've hopefully now convinced you and hopefully brought out that same feeling of inspiration, that same feeling of, hey, the work that we're doing in this class is relevant and important by presenting a cool concept and a cool application from each of the units that we've scheduled for the course. I think Dr. Huth will have his words at the very last day of class, but because I won't get to have the penultimate day, I'll go ahead and say my piece now. You guys have learned so much important stuff. I'm really happy for those of you who got to TA for Neural 1, it's been an honor to TA for you guys again. I've seen some of you guys grow in ways that are more so than I did. I was a slow grader then, I'm still a slow grader now, but I hope that you guys walk away from this class. I know, even if you guys don't remember how to index and slice into an array 5 years from now, I hope that you guys remember that the work that you put into this class was meaningful and important. It's been an honor to TA for you guys again, and I'm really happy that you guys have learned so much. Any questions before we wrap up? Okay, that's it. Next Friday is going to be finishing up PCA, and then Monday is going to be just review for the final exam, right? All right, that's it. Well, thank you guys so much. Of
Thanks, Henry.
course. Right.